Okay. So again, if you want to get lost in your microscopes while I'm chatting, that's fine. But if you hear, if you hear me actually say something that sounds relevant, I would appreciate you tuning back in. So we are going to cover 4 types of tissue. They're the 4 main types. They are connective, muscle, nervous, and epithelial. Each of these has subdivisions. We'll get to them as they become important. Remember your basic definitions of biological organization. Cells organize into tissues. Tissues are a group of cells that together perform a specific function. Organs are the next higher order. They're composed of different tissues that create some higher order function. You know all the familiar examples, but today we're focused on the tissues. So, connective tissues, epithelial tissues, muscle tissues, nervous tissues. Practice, practice, practice. Okay, so one by one. Epithelial tissue. We've already tackled this. We've seen 2 different epithelial tissues in the frog gut in previous discussions. One was on the inside, one was on the outside. And these kind of represent 2 of the kind of functions you get with epithelial cells. First off, they are tightly packed. In what way are they tightly packed? Each cell is nicely abutted next to the other, and the cytoskeletons are actually connected through desmosomes touching between their cell surfaces. They also have gap junctions and all sorts of things just meant to seal up that surface. So one cell is very tightly knitted to the next one. That leaves most of their functions are either absorptive or protective, and for that purpose you need a free surface. So in the inside of the gut, that is the free surface of that epithelial tissue. On the outside of the gut, that's the free surface that's protective of the whole tissue. The inner lining of your mouth is another of these free surfaces. So when you have a nice sheet of cells, operative word, sheet of cells for epithelia, remember that they form surfaces. Because they are so into binding to each other and creating these surfaces, they don't really have much room for the irregularities that are caused by vasculature. So they do not have access to their own blood supply. They get it indirectly through the connective tissue that's underneath it. So 2 other places you find them. Skin is going to be one example of an epithelial tissue. That's the epidermis, not the dermis and the other parts. And lining of almost every major organ. So we already covered that they're protective. They secrete things typically through goblet. They typically contain goblet cells which secrete things like mucus, and they can also go the other way and absorb. We see that in the gut. There are 3 different factors that you use to describe these things. If they come in how many layers they are. Simple means 1 layer. Stratified means several layers. Pseudostratified means it's actually 1 cell layer thick, but it kind of looks like there's multiple levels. And those multiple levels, well, they look that way because the nuclei are distributed unevenly, and the cytoplasm is distributed unevenly. Typically what's happening there is the nuclei are moving up and down according to their cell cycle. You see something like that in the developing nervous system. Their shape is extremely descriptive, except for squamous. Squamous, equate that with squashed. So a squamous epithelium is these cells that distribute their cytoplasm along the flattest possible surface. Things like the inner lining of your blood vessels, lining of your major organs like the lung, so forth. Cuboidal, you have a nice regular pattern. It is roughly like a cube. And columnar means they're actually stretched north-south. And I don't think we need to discuss combining those terms. The examples are listed over here and illustrated. They're quite nice. Connective tissue is far and away the most diverse of these. It contains things that you would not think it contains. It contains blood, it contains bone, everything, tendons, ligaments, the fascia that holds your organs in place, all the kinds of things that hold the vasculature that supply every other tissue. All that's connective tissue. The biggest surprise in there are blood and actually fat are both connective tissues. So these things are best at making other things stick to each other. They do that by secreting, I don't know when I'm supposed to cover this, but by secreting extracellular matrix. So when you look in a microscope and you see that there's not nuclei in any kind of dense pattern, but you see that there's a bunch of fibers running in between cells, that's one of these patterns you should pick up on that says this is a connective tissue. So they secrete all sorts of these extracellular matrix proteins, collagens, and the like. So what? You find them everywhere. I'll just list joining other tissues. The skin is a great example of this. When you're looking at your skin, see if you can identify the epithelium that is the epidermis. It's really only a cell layer or two thick. But underneath that are several layers of connective tissue that get the various jobs of the skin done in the dermis. So there's the stuff that holds the epithelium down, there's the stuff that vascularizes it, there's the fat that insulates, and so forth. It functions to support or protect. Think of bone, that's your major source of protection. Support, that's what the skeleton is for. Locomotion. Things like tendons and ligaments connect your different muscles to the structure that makes them work, so that's all relevant. Transport is your blood, storage is your fat, and your bones. Why is bones storage? Stores calcium. Outstanding. So there's that, and we've listed defense, that is the formation of the white blood cells in your bone marrow. Okay. We will spend a lot of time on connective tissue. You should be able to look at a typical picture. These are all of our samples and identify them. So H&E, the pink staining here, is helpful for this purpose. You'll notice labeled here fibroblasts. That's really just kind of the space in between here. And all this secreted stuff is collagen, so that's something like a tendon or a ligament. You'll find something loose like this, where you can see the fibers running in each direction. Something like fat here, which the major part of the cell is just a big fat globule. Bone, which we'll get into more on the bone section, but it has living cells embedded in a calcified cartilage base. And then blood cells. Please don't get this wrong on the test, by the way. Which of these are the red blood cells and which of these are the leukocytes, the white blood cells? And what gives it away? Class? The one that's stained are leukocytes. Red blood cells are enucleated. They dispose of their nucleus for the rest of their life. That's why when you do this nuclear stain, they don't stain with it. Moreover, they kind of have this dimple-shaped shape. So if you see cells with nuclei in the middle of your blood sample, you know that you're talking about a white blood cell. Okay? So I think we discussed these. Muscle, we will grill you again and again, be able to identify all three types of muscle. So major characteristic is it's capable of contraction. It gives us movement. That's its purpose. You find it all over, either in skeletal muscle, in blood vessels, making your gut move, and so forth. And please refer to this slide early and often. It gives us the major distinctions between the three types of muscles. There are skeletal muscle, smooth muscle, and cardiac muscle. So let's create a hierarchy here. The smooth muscle is all by himself. It does not have striations because these lines, these striations that are set up by the sarcomeres, they're actually the kind of the whole motor complex that make the muscle able to be contracted. In smooth muscle cells, it's just organized in such a way that you don't see it. in striated muscle, which is skeletal muscle, and cardiac muscle, both. These sarcomeres are visible as these lines. The major thing that distinguishes cardiac muscle for the heart from the skeletal muscle, which is for your locomotion, is two things. First is cardiac muscle is highly branched. They do this so that the cells can communicate to each other, so that when a calcium wave comes and it fires that muscle cell, the whole heart beats in unison, or at least in its systematic pattern. So for that, you need to have the muscle cells connected to each other to form a web, and so they do. The other thing is these intercalated discs, which is the main thing that you'll be able to tell under the microscope itself. This is the most obvious thing. Do not leave today without identifying an intercalated disc to me or somebody else, and be very sure about it. Okay. Nervous tissue typically doesn't get much treatment, but remember that this is a way our body communicates through electrical signals. It generates these action potentials. That's your keyword. That's basically a difference in ion concentration that gets equilibrated when we get activated. That's a rapid way to communicate that signal. We find neurons and their support cells all over the body. Interestingly, there's more neurons in your gut than there are in your brain. Think about that. So if anybody ever thinks with their gut, you have a conversation starter. Okay, as we learned from the TED Talk, most of what this is about is making our body move and processing why you would want to. When you look at your tissue, what tissue do you have for the nervous system? The spinal cord? Yeah. So you might as well make a study of it now. There's kind of a regional thing that you can distinguish on the spinal cord, and that's gray matter versus white matter. So the white matter is actually mostly the cell bodies of the insulating cells, so it's got a lot of fat. That fact by itself, I don't know if it would make sense to you, but here's how it works. You've got an electrical signal. Electrical signal in a body is not terribly different than an electrical signal running through the wires in your house. You need insulation so that it doesn't diminish its intensity or cause something else to receive that electrical impulse. So to keep these things properly, to keep these signals going properly to their signal, and also to make it more efficient, there's these wonderful cells called Schwann cells that wrap around the axons of each neuron that help insulate it. And those Schwann cells essentially just carry a bunch of fat to insulate it. Those Schwann cells are one of the types of neuroglial cells. I say neuroglial just to be courteous because neuroglial is in the slide. Everybody just calls these glial cells. Also, they're largely overlooked, but they have all different types, probably more diverse than neurons. There's astrocytes, there's oligodendrocytes, there's Schwann cells, and so forth. All carrying out their own function, but all those functions together essentially support the activities of the neurons. When you're looking in your microscope, you can tell them apart because they're small and they don't stain these nice fibers like the neurons do, with H&E that is. So a neuron, you're going to look, it's going to have a big fat cell body. And it's going to have pronounced fibers coming off of this. Some of them are one of them is an axon. Several of them are going to be dendrites. Axons send the signal, dendrites receive the signal, by and large. Sorry, just let me finish up the gray matter, white matter. Gray matter is actually the place where most of the neuron cell bodies live. Okay? And so the neuron cell bodies live in one place. Their actual fibers, insulated by myelin, in the white matter, live in a different place. So if you think just real quick about your spinal cord, which is essentially just the axons running from your brain down to communicate to the rest of the body, there's not a lot of nuclei going on in the white matter that you see in your slide, because it's all the axons bundled into these sheets. So it's like a whole bunch of fiber optic cable just laid down together. Interestingly, the white matter and the gray matter are inverted spinal cord to brain. So I have to remind myself in the spinal cord, the gray matter should actually be on the inside and the white matter should be on the outside, if I have it right. But I will correct that if I'm not.